Good morning to everyone. The Biochemical Society and Portland Press are pleased to welcome you to this webinar, which is a part of our Biochemical Focus webinar series. Topics in this series include different research areas in molecular biosciences, as well as the practical sessions to support career development. Each webinar will give you the opportunity to ask questions via text, and we welcome suggestions from you for future topics and speakers to feature in our webinar series. Please see the website for more information. I am Irina Tikhonova, and I am reading molecular modeling from Queen's University Belfast, and I'm delighted to chair this session. And this webinar is dedicated to computer-aided drug design, types, uses, and opportunities. Our invited speakers will be exploring different computational tools, applications, and software. And also we will see how computer-aided drug design methodologies can quickly become important tools in modern drug discovery and development. Before I hand over to our first speaker, I would like to mention that, again, questions will be asked at the end of the webinar, but please do send your questions during the talks. If you have a question, please type it in the question box as shown in the image on the screen, starting who your question is for, and we will try to answer as many as possible at the end. And now I'm delighted to introduce to you our first speaker of today is Professor Abdulilah, an associate professor of chemistry at Biruni University, Istanbul. Professor Edger has background in organic, computational, and medicinal chemistry with specific training and expertise in computer-aided drug design tools. He established his lab at Biruni University in 2014, and his group focused on earlier and mid-stage in silico drug discovery that encompass both ligands and structure-based design. He's supported by leading software companies in the field and collaborates with national and international researchers. He uses computational tools to address various drug discovery challenges and also provides structural insights for experimental work of medicinal chemists, organic chemists, pharmaceutical chemists. So today he will give us introduction to computer-aided drug design methods. Abdulilah, please.
Thank you for the introduction. Hello, everyone. It is great to be here, and it is my pleasure to introduce you a basic introduction to computer-aided drug design methods. Okay, let's begin with the simple drug design processes, simple steps involved in drug design. The first step involves identifying the cause of a specific disease. The next step is to search for the target that is involved in the disease. You have to be careful selecting the correct target because some enzymes, for example, are closely related, but they are not identical. They have isoenzymes, they are isoenzymes, they have different subunits. For example, phosphoinositide has 4 different subunits, alpha, beta, gamma, and delta. If you are interested in anti-inflammatory effect, you have to select the gamma type. So selecting the correct target is also crucial here. The next step is to find out the lead compound. The lead compound is a compound that has a specific activity, even low, but it is a starting point. The next step is to optimize the target, the lead molecule. Optimizing the target, the lead compound means to increase its activity or to decrease its side effect toxicity. And the next step is clinical trials, phase 1, 2, and 3. The next step involves the approval process. If you are lucky enough, then your compound, your drug candidate becomes drug available at the market. So as you can imagine, based on the literature data, it is provided by 10 drug companies considering the discovery phase, clinical phases 1, 2, and 3, and FDA approval process. Drug discovery costs more than $2.5 billion to develop. So drug discovery is really time consuming and it is cost intensive. That's why new approaches are needed. The new approach here is the cutting edge technology here is the computer-aided drug design. It involves many branches of sciences, including chemistry, biology, medicine, biochemistry, pharmaceutical chemistry, organic chemistry, pharmacology, even physics, and many more. And we can divide computer-aided drug design into two broad categories. If 3D structure of the target protein is unknown, then you are left with the ligand-based drug design tools. If the active compound is unknown, then you search by, it is called random screening. You don't have any idea, you just screen randomly. But if you have an active compound, you use similarity search to find out a compound that has similar structure and similar activities. And Pharmacophore maps, QSAR studies, combinatorial libraries are all involved in ligand-based drug design. Structure-based drug design is very crucial in the drug discovery process. If you know the 3D structure of the target, you can just design your own compound from the scratch. It's called de novo design. Unfortunately, the success rate is not too much here, not too high here. But the most commonly used techniques are molecular docking, molecular dynamics. They are all structure-based drug design. So let's start with the QSAR. QSAR is the abbreviation used for quantitative structure-activity relationship. Studies based on the relationship between structure and the ligands and activity are called SAR, structure-activity relationship. If you try to relate those relationships, formulate those relationships with using mathematical algorithms, formulas, then it will be called QSAR. So it means you are relating those relationships using numbers. Probably the first general formalization in QSAR was reported by Crum, Broman, and Fraser in 1868. They reported that biological activity is related with the function of properties, parameters, descriptors of compounds. The modern QSAR technology started in 1964 with publications by Hansch and Fujita and Free and Wilson. Parameters are anything that is related with the biological activity that can be represented by numerical values. It can be fingerprints, electronic parameters, hydrophobic and steric parameters, and many more parameters can be involved in those sections. If you don't have any hypothesis that helps you in selection of a good descriptor, then you have to use multivariate methods in which you are trying to find the best descriptor that is related with the biological activity. Statistical tools are needed here. For example, multiple regression analysis are used, partial least squares are used, but with the development of computer technology now, we have artificial intelligence that helps us to find out good correlation, more precise, more accurate relationship between parameters and the activity of the molecules. Machine learning, deep learning methods are used, and you can find many publications nowadays that uses the keywords artificial neuronal networks or deep neuronal networks. It is a simple example for QSAR. We have a set of molecules here, and the effect of R and X substitutes are investigated here. And you have to divide your compounds into two sets. One is called training set, the other is called test set. Training set compounds are used to train your model, while the test set compounds are used to validate your model. So you have to use descriptor calculation softwares. Here, more than 200 descriptors were calculated. And nowadays, there are software available at the market that can calculate more than 5,000 descriptors that are 2D descriptors, 3D descriptors, etc. Some statistical methods were used here, and then you are you are generating some equations here. PIC50 is the biological activity of the molecules, and we have some constants here. The red ones are the parameters, and those are statistical parameters. Each parameters should be in acceptable units. For example, we have correlation parameters here and cross-validation parameter here, and all those are important to validate your model. If your model is validated, then it means if you are you have an experimental unknown compound, you don't have any idea of the biological activity, just calculate those parameters here and put it in the equation. It will give you a theoretical biological activity. So that's why those data used in generating a QSAR model is very, very important. The more reliable your data is, the more accurate results will be. Pharmacophore studies are, again, common use methods, and pharmacophore groups define important functional groups that play a crucial role in the interaction of a drug to its target. We have 2D pharmacophores and 3D pharmacophores. 2D are simple pharmacophores. They define the minimum skeleton connecting important binding groups. For example, in morphine, the important group for energetic activity is those nitrogen and phenyl groups. You connect them together, and it gives you 2D pharmacophore skeleton here. But if you look at the similar compounds like metazocine, levorphanol that has similar activity, you will see the same pattern here, same pharmacophore group here, nitrogen, phenyl group, aromatic group here, nitrogen connected to the same group here. So we have same skeleton, so they have similar activities. But of course, we are living in 3D world, so 3D pharmacophore is important here. They are called pharmacophore triangles. Here, the pharmacophore groups are connected in 3D. It means you have to define the bond length, bond angle, bond torsions here. And if, for example, hydroxide group itself is not so important here, but hydrogen bond donor property is important, so you just define it as hydrogen bond donor or acceptor feature. So as such, amine group, if it is not so important, the basicity is important here. Just define it as basic group. Any basic group here, any hydrogen bond donor or acceptor here, and here, any aromatic group here can work as your active compound. So you connect them in 3D. It will be your 3D pharmacophore group. You can generate a pharmacophore from target binding sites. For instance, here is a binding site here. In this amino acid residue, we have carboxylate anion. So a ligand which has a positive group can interact with this negative group. We have hydrogen bond donor and acceptor feature here. So any group that has this property can interact with this one. Any group that can interact with aromatic group can interact with this phenylalanine here. So you define those points together, and you generate your pharmacophore group from the target. Okay. So one of the most common used method is molecular docking. It is simply fitting of a drug of a compound into the binding site. It considers the size, shape, the binding interaction of the ligand and the active site of the target. Okay, so those docking softwares generally gives the best poses based on the energy values. So it is called scoring. It depends on how well they fit and how many binding interactions are formed. Simply to put, the formula is that the software calculates the Gibbs free energy of the free receptor. It calculates the Gibbs free energy of the free ligand, and also it calculates the Gibbs free energy of the whole system. If the ligand's target interaction is favorable, then we expect the energy value to be low. So the resulting Gibbs free energy of the bonding should be negative. The more negative the value is, the higher the binding, biological activity will be in theory. Okay. Molecular mechanics and quantum mechanics can be used here. Molecular mechanics follows the simple laws of classical physics. It ignores the electron interactions, and the molecule is treated as spheres connected by springs. But in quantum mechanics, it considers the interactions of the electrons too. For simple molecules, it makes wise to use quantum mechanics, but for larger molecules like proteins that contains thousands, 10,000s of atoms, like 30,000, 50,000 molecules, so you can ignore the interaction of the electrons. Sometimes hybrid methods can be used. The inner part of the binding site amino acids the high-level base sets are used here. The nearby atoms are treated with the low intermediate level basis sets, but the rest of the macromolecule can be treated with the molecular mechanics system here. There are a number of papers published, but unfortunately the success rates are not so good. So simple molecular docking is simply better. Sometimes we treat target and the ligands rigid. Sometimes the target, the protein, is being treated rigid, but the ligand is treated as flexible. The most commonly used method is the second one. It is more complex but feasible. If you treat both target and ligand flexible, it is very complex and expensive on computer time, but the success rate on the second one is better than the first and the third one. Sometimes we use induced feed docking here, as for example, a software tries to fit the ligand in the binding site, removes the amino acids to make the fitting process better, and then it generates amino acids again. It calculates the binding score, and then it tries another conformation, puts into the binding site again, removes the amino acids, and then generates the amino acids again, and then calculates the binding scores. It is called induced feed docking. We have some software that has this capability. Those are main steps involved in molecular docking. First, you have to prepare the ligands and macromolecule. It is one of the most crucial part in molecular docking because macromolecule, the protein, should be checked for errors. Sometimes amino acid residues are missing. You have to check for the bond orders. Hydrogens are missing. You have to optimize, minimize all your protein here. And ligands also should be prepared for covering a large chemical space. You generate many conformations here. You consider the ionic states here, different ionic states here. You consider tautomers here also. And then, of course, you have to identify the binding site of the protein. Then the software samples ligand macromolecule complex. At the end, you have to analyze the results in 2D and also in 3D. Okay. All those interactions are based on hydrogen bonding, salt beaches, hydrophobic contacts, metal coordination, and many more interactions are considered here. Of course, molecular dynamic is a must in many cases because the docking might be an instant picture of your case. So you need to see whether those interactions are stable or not, whether the protein is stable upon the binding with the ligand. That's why we try to prepare a system. As you have seen in simple molecular docking, we don't treat water molecules. We generally remove the water molecules. But in our biological body, we have many water molecules. Also, we have salt concentration. So we mimic the physiological condition. We prepare a system. Here is a picture of a prepared system here. Here is our protein here, surrounded by a water molecule. So we try to mimic the physiological condition here and try to simulate all the system like a movie. Okay, then we observe the movement. We observe the dynamics of the proteins here, whether the protein remains stable, whether the ligand remains in the binding pocket or not. Sometimes it can be diffused away from the binding site. That's why this molecular dynamic is very crucial too. Here's a closer look into the binding site of the molecular dynamic picture. As you can see, the sample ligand here is being like grabbed with the binding site. Okay, so you can see the interactions, how long those interactions exist here. And as you can see, the lower part of the ligand is being sticked in the binding site. It does not diffuse away from the binding site. And during whole course of the simulation, a metal coordination, zinc metal coordination remains constant here during whole time of the simulation. Okay, so that's why the dynamics, to analyze the dynamics of the protein, analyze the stability of the interaction, it is important to carry out molecular dynamic simulations. So you can analyze the protein root mean square deviation and ligand root mean square deviation throughout the whole simulation. Okay, here is an example for 200 nanosecond of simulation. Here we expect the protein RMSD value to reach equilibrium at some point. As you can see, we have some fluctuation here. The blue one shows the protein RMSD, and after about 50 nanoseconds, the protein reached at equilibrium, and ligand fluctuation should be low again. Okay, so you can see the fluctuations of each individual amino acids here. So the green vertical lines represent the amino acids that are interacting with the ligand. If the fluctuations here are low, it means the amino acid is being stabilized by the ligand upon the binding. As you can see, in many cases, upon the binding ligand, upon the interaction of the ligand, the protein is being stabilized. We prefer the whole RMS F value to be lower than 2 angstrom, but in my opinion, do not stick with 2D results. Always try to see the ligand RMS value might be, for example, very high, but if you look at the movie, you can see whether the ligand is really diffused away or the RMSD is because of the flexibility of the bones, some rotatable bones. So it is better to combine those data together. And you can see in 2D how long an interaction exists during whole course of the interaction. For example, we have hydrogen bond interaction here that lasts for 90% of the simulation time here. We have again pi interactions here that last for over 50%, over 70% of the simulation time. Okay, so let's start with the visual screening. It is simply searching potential drug candidates to databases using queries. You can collect synthetic compounds, natural compounds, convert them into 3D, you sample all the ligands, you try to generate all possible conformation ionization states. For example, here is a ligand, but after the preparation step, you can end up more than 1 or 2 conformations, sometimes with more than 10 conformations. You can generate R isomer, S isomer, E isomer, Z isomer, different ionization steps to cover large chemical space here, and then you prepare your database, you search through database, and the results will be your hit compounds. Upon studying, analyzing on the hit compounds, optimizing on the hit compounds, you can find your lead compounds here. Okay, let's start with an example here that I used to combine many tools together to publish this paper. Here's my Pharmacophore model in 3D. We have hydrogen acceptor, hydrophobic regions, ring aromatic polarizable regions here. Of course, in each step, you have to validate your model. For instance, in virtual screening, you have to prepare a known database to calculate enrichment factor, for example, or bedrock parameter, or robust initial enhancement to see whether your Pharmacophore model is capable of picking up the correct active molecules or not. You prepare, for example, 3,000 inactive molecules, you put 100 or more than 100 active molecules in the prepared database. You first try to test your model. If it passes those parameters, then use these parameters into screening of large chemical databases. So in my database, I had 7 million compounds with more than 150 million conformations. I used some filtering here and some starting with the simple to the advanced docking techniques. I filter I filter my compounds. 10% of the successful candidates passed through the other filtering methods, other step. So 10% from standard precision docking were subjected to extra precision docking. Normally scoring functions are primitive, so I used more advanced, more rigorous scoring function like MMGBSA. At the end, I had 361 hits. After the ranking and visual inspection, I selected 15 compounds. Okay, here's another example. If you see after the docking, if you see the 2D ligand interaction diagram, you might think that hydrophobic interaction is important here. So increasing hydrophobicity should increase the biologic activity. If you remove the hydrogen here, replace it with chlorine and fluorine, hydrophobicity increases, but unfortunately, after the experimental results, we saw that activity decreases. So 2D analysis is not enough, so you have to analyze carefully by visual inspection in 3D also. If you look at the 3D, you will see that there is actually a T-shaped pi-pi-stacking interaction here. So as you may know, in aromatic rings, we have electron density above and below of the plane, and at the edge we have a partial positive charge. So here we have negative side, here we have positive side here. So the more negative, lower electron density here is important. If you increase the electron density, it will decrease the interaction here. That's why if you generate the electron potential surface around the each ligand, you will see the red color represent the high electron density region, blue color represent the low electron density region. As you can see, the lowest electron density region belongs to the one that has no substituent in this aromatic ring. If you replace it with chlorine, fluorine, and electronegative atom, it draws electrons from the system, making this high electron density region. So that's why biological activity is decreased. So let's summarize it. Computer-aided drug design consists of many tools. I suggest you to use different tools to combine different tools to obtain a more accurate results. So here you can use molecular mechanics techniques. Sometimes quantum mechanics are used. You can calculate ADME properties, pharmacokinetic properties. Using statistical parameters, you can validate your results. You can use standard docking methods, extra precise docking methods, molecular dynamics. At the end of all those tools, you will end up with some hits, and you can go further with the wet lab experimenting. But be careful. Always, each step should be validated before predicting any property or a complex system. For instance, internal validation should be carried out in molecular docking. In 2D or 3D QSAR, cross validation, Fisher validation, student t-test, some other tests should be done. Okay. In virtual screening, again, enrichment factor, robust initial enhancement, bedrock analysis should be done to validate your system. Okay, I have only 1 minute, so that will be all for this section. Thank you. I'd like to thank Dr. Rita Ponzuna, Sean Ollinger, and I'd like to thank those software companies for supporting my research group, Schrödinger, Spartan, Gaussian, Asset, and Wally Macmillan International Higher Education Groups, and Dell Technology for supporting my group. And once again, I'd like to thank Biochemical Society for their nice invitation, the group members, the team members, and all members of society. Thank you.
Thank you, Abdulilah. That was a great introduction. And now we move to the next speaker. So our next speaker, invited speaker, Dr. Lampros Milanos, a senior computer-aided drug design scientist at Charles River Laboratories. Lampros has been working for Charles River for 3.5 years now as a senior CADD scientist. So he's based on Chesford Research Park near Cambridge. His career start was 8 years ago as a medicinal chemist, and he followed career opportunities that made him a computer-aided drug design scientist. Today, he will discuss his own career path and explore different opportunities available both in academia and industry. Lampros, please.
So yes, hello everyone. I would like to take this opportunity and thank Biochemical Society, Portland Press, and the organizing committee for this invitation. It is my pleasure to be here today. So I have prepared a presentation discussing my career path, my current role, responsibilities, and a couple of slides discussing drug discovery industry and opportunities that can be found out there. So first of all, I'm Greek. I grew up in Athens. Most likely you know us for our touristic destinations, delicious food, and our sirtaki dance. Just a small advertisement of my country. But I would say let's get more serious now. So my career path, I studied chemistry, so Bachelor's of Science at the University of Ioannina in Greece. And two subjects particularly took my attention. It was organic chemistry and biology. So ever since I tried to combine these two subjects, starting with my future studies. So medicinal chemistry was a very nice, let's say, topic or discipline that combines both. So I moved to Glasgow to do my Master's of Science at the University of Glasgow, and then moved in Germany for my PhD. I spent 3 years there, where my at the time PI was Dr. Nusrat Sharma. The PhD program started with a medicinal chemistry approach. Initially, it was planned to be a medicinal chemistry program, but along the way, there was an opportunity for us to investigate our results in silico. So I moved to a computational group, more specifically Professor Tim Clark's group, and we did some computational work as well in my PhD. So after that, I was very keen on combining computational chemistry into my research, as it was providing rational design to the ligands I was designing to make in the lab. So I found an opportunity in New York at the Memorial Sloan Kettering Cancer Center. My PI at the time was Dr. Gabriela Kiosis, and that was my first postdoc, and we started with a computational chemistry approach to try to answer some limitations of the current of the project at the time, and find solutions and then provide prioritization for compounds that would be promising for our target, and then moving to the lab and synthesizing these compounds. After this postdoc, I moved to Italy at the Italian Institute of Technology as a senior postdoc. And then I would say you can see the person that is our opposite. It was more of a medicinal chemistry, but again, I always tried to combine both disciplines into my research. And yes, both medicinal chemistry and computational chemistry at this postdoc as well. And then found an opportunity and I would say Charles River offered me this to become a pure computational chemistry scientist and be part of the CADD team. So I've done also a few more placements along the way during my studies and my career. So Denmark, in Italy, and Greece. And then briefly describing what Charles River is, what kind of company. So it's a contract research organization. It is a global CRO, and the last 5 years has supported more than 80% of the FDA-approved drugs. So Charles River covers all stages of pharmaceutical research, starting from basic research to commercial manufacturing. My department belongs to the discovery services that cover all early drug discovery and non-clinical development stages, and some aspects of the clinical development. So I am a senior scientist too, but overall, Charles River CADD scientists are responsible for supporting medicinal chemistry teams by providing help to prioritize compounds. So we usually get a set of ideas, and we are using a variety of computational tools and techniques to prioritize and suggest the most promising ones to accelerate the efforts of the medicinal chemistry team. In addition, we also suggest our own ideas using idea generation CADD methods to design compounds based on a rationale. So there are various techniques you can do that, from library enumerations to even having an idea and then draw it on ChemDraw, and then do some modeling to identify a potential, for example, docking mode. We support structural biology by performing quality controls on the X-ray structures before release to the client. So that involves a multi-step process using a variety of tools, and we perform diverse analysis focusing on the ligand region. We support internal efforts to develop CADD methodologies. So it is important for us to establish workflows that can support our projects, our clients. And then we have also the design of compound libraries, helping efforts to design this type of libraries that could target a particular family of proteins, for example, like kinase. We also evaluate CADD technologies, so we always want to be up to date, and we evaluate latest. We follow latest developments by evaluating tools and approaches that we think that can be useful for our projects and clients. So eventually, when we do the assessment, then we decide if we want to purchase or not. And also we have the project meetings where we present to CADD-focused audiences, where the presentation can be more technical, but also we present to diverse scientific audiences where we have to be we have to present and keep it simple, let's say. So we are involved in all early drug development stages, from hit identification to lead optimization, and every stage may require a slightly different strategy or a significantly different strategy depending on where you are and what you want to achieve. And every step can also involve a different set of tools in order to achieve your goals. So as a discipline lead, I'm responsible for the development of the CADD strategy, managing the CADD resources, and or executing the experiments, the CADD experiments. We are responsible for presenting to the client. Usually we have multiple projects, so as you can understand, we tend to have regular meetings with clients as well as internal ones. Last but not least, we are responsible for all communications involving the CADD discipline, so everybody is up to date with what we are working on, the strategies we are following internally as well as externally. Everybody is aware of what we do. We try to be as transparent as possible, and we agree on the strategies designed to support the programs, the client programs. So I think we discussed the types of CADD approaches found that has been used in CADD. So I will keep it brief. This is the CADD approaches we also use in our company. So structure-based approaches, including docking, MD simulations to free energy perturbation calculations. We have the ligand-based approaches focusing entirely on the ligand. So this aspect involves analysis or modeling of the ligand, such as conformational searching, Pharmacophore modeling, QM calculations, etc. And we also contain a proprietary heat expansion platform where we use query molecules to expand heat matter, as well as to develop SAR around a specific chemotype. And then we have the AI in chemoinformatics. We contain an internal toolkit for idea generation, clustering, structural alerts, and much more. We have available our internal global models, as well as we develop local models for particular projects when necessary. We are working on our generative AI modeling workflow as well. We offer CADD-based plate selection, so it is a bit more focused, high throughput screening, using plates with properties that are suitable to the target and client needs. And some methods here I'm illustrating. We have the PhysChem property prediction, ADME, predictive models, generative models, etc. So this slide describes types of industry in drug discovery. So we have pharma and biotech or startups, and then the contract research organizations such as Charles River. So pharma and biotechs usually run their own internal programs. Larger pharma are able to accommodate all preclinical drug discovery stages internally, as they have the capacity and the facilities to do that. Smaller biotechs may partner, sell, or can be bought by the big guys. Some may outsource some or all of their research to CROs, and there are organizations that are purely virtual and outsource all of their wet work to CROs. So this type of organizations tend to focus on a particular disease, on particular disease areas. Bigger pharma companies may have a set of departments focusing on particular disease areas, for example, oncology or inhaled diseases. On the other hand, contract research organizations provide research services to clients. We have all types of clients, can be an academic group to a pharma and everything in between, biotechs, non-profit organizations. So everybody is welcome to us and to CROs. CROs offer multiple business models. So FD-based models, fixed cost, success share, achieving milestones. We have programs or clients requesting research or focusing on a particular discipline. For example, you may have a CADD standalone project, or very often we have clients requiring or coming to us with an integrated project, so that involves multiple disciplines. So CROs usually work on diverse range of projects. We cover a variety of target classes, diseases and route of administration. So everything, of course, is based on clients' needs. And here I have a slide where I give an overview of potential opportunities you may find in CADD. So classify them based on type of institutions. It can be in industry or academia. In industry, you have the typical drug designer positions, from scientist all the way to research leader, depending on your experience and what is suitable to your background and experience. Yeah, we have the method developers, focusing on developing computational approaches, methods, and tools. We have application scientists, where they work for companies that sell platforms and softwares to their clients, and application scientists are there to support with anything scientifically they require. We have the technical specialists, AI scientists that become more and more popular these days, cheminformaticians, and then you can move to not so much pharmaceutical based positions, such as a journal editor, where you can work for a journal covering computational research. You can have, there are also environmental scientists involving computational chemistry, and then you have also the management position. So if people are interested also in management, they can move to the management ladder and manage their own group. So we have then the academia, very well-known postdoc positions. Postdoc position for computational chemistry contain method development, focus position, drug design. You can have positions focused entirely on quantum mechanics, even sustainable energy. So a lot of things that can be found out there. There are technician positions that may also not require a PhD. Maybe having a Master of Science can be enough to have a position like that. We have permanent staff positions, usually senior staff scientists. I would say these are few and highly popular. Cheminformaticians in research groups, usually these positions are permanent as well. And then we have professorships that requires you to earn your own grants for your research and team. And after some years, yeah, the position becomes tenure, so permanent. So yeah, thank you very much. I think that was my last slide. I will be happy to answer any questions you may have.
Thank you, Lampros. That was a great talk as well. So indeed, now we open for questions for both speakers. So if you have question, please type it in the question box as shown in the image on the screen. So, okay, while we're waiting for questions, I would like to start, and I would like to ask a question to Abdulilah. So, well, I guess, Abdulilah, you gave a great overview, and also in the last slide, you showed software that you use. But anyways, could you just recommend what kind of software would be good to use, commercial software, or in fact, open source, if someone wants to start to do computer-aided drug design project?
You mean the open source freely available software or any software?
Well, I guess, yeah, you could comment on both of them.
Okay, actually, I used both. I used different software in the past. Now I'm using Schrödinger. Unfortunately, they are now supporting, and I keep on continuing with Schrödinger software. I used open source, freely available software too. It actually depends on the case. And the free software are hard to use. They are not so user-friendly, and you are limited with some modules. But the licensed software, you have many models there. You have, for example, docking there, molecular dynamics there, the model that you can use to calculate the pharmacokinetics, and many more models are in this software, in one software, and they are user-friendly. And which one is the best? There's no such thing. Don't believe such things. But I'm happy with the Schrödinger software, but it depends on your specific case. For example, in molecular docking, we have different settings, different methods. You have to apply all those settings, all those models, stick with the validated, accurate model in your case. Sometimes induced docking will give you very good results. Sometimes simple standard molecular docking will give you very good results. So always stick with your case that has been validated by yourself.
Okay, thank you. And then we got first question for you, Abdulilah, and the question is, are the neural networks used to optimize molecular docking generally open source or widely published?
Excuse me, come again?
So are the neural networks used to optimize molecular docking generally open source programs?
In open source, Autodoc Vina is widely used. In open source, freely available, Autodoc Vina is widely used. I published using Autodoc Vina a few years ago.
Okay.
And some software, big software companies can give you trial license, but some companies don't allow you to publish with the trial licenses. But Schrödinger will allow you. They allow you to publish with academic trial license. So you can just contact them.
Okay, so thank you. Another question here about ligand-based pharmacophore modeling and structure-based pharmacophore modeling. So which techniques most useful?
Okay, actually, there's one more thing that combines both structure-based and ligand-based pharmacophore model, which is called energy-based pharmacophore model. For ligand-based pharmacophore model, it depends on how good your ligands are, because ligand-based pharmacophore model are generated using a set of available compounds. So if you have good set of available compounds that have good biologic activities, and if you rely on those data, then you can use ligand-based pharmacophore model. Of course, in theory, structure-based pharmacophore model should give you better results because it is generated using the target itself. But the ligands, you are generating using the set of ligands, you don't know their active conformation, you just stick with the active molecules, and inactive molecules you generate using all those limited data. But structure, the active conformation of the protein can be available in protein data bank, so it should give you better results.
Okay, thank you. So we actually have quite a lot of questions, so we'll try to address some of them. And I think now there is a question for Lampros, and the question around, just one moment, I already missed it a little bit. So how would you describe your experience in academia versus industry? Also, how do you use generative AI in industry projects?
So I would say both were interesting. I enjoyed both. In academia, I've been to some places, had the opportunity to travel a lot, interact with a variety of research groups. I joined several symposiums and a lot of collaborations as well. So the only caveat I would say is the fixed term contracts. So eventually that you have to find a research group that can provide you a contract for a bit longer term, or every time to find find grants to fund your own research, or at least your PI to find grants to fund your research or the group's research. So overall, I would say that's the most challenging thing. But overall, it was very enjoyable experience. I had the chance to work for many groups and interact with many people. On the other hand, we have industry. I would say it requires a bit more of organization, and especially in CROs, because we are dealing with clients. Everything is about the client needs. We may handle a bit more workload compared to academia, but then we have the permanent contracts, so we don't require grants to fund our research. And actually, another difference is that usually we don't publish so often compared to academia, as also it's not so necessary in industry. But I would say both were very enjoyable experiences. Eventually, usually this is something common. You start from academia, and then when you find an opportunity that suits you, you move to industry. There are, of course, some examples that moved from industry back to academia, but usually this is the trend. And the second question was about generative AI in our projects.
Yeah, so it was about, one moment. So it was indeed, so the future of generative AI and how do you use an industry?
Yeah, it started becoming more and more popular. So we have AI specialists in our company developing this AI tools that, for example, deep learning generative models that are able to generate ideas meeting all the parameters set at the beginning of the in silico experiment. So it can be really useful for a client. We have the medicinal chemistry team, but we also need to have a way or approaches that we can suggest our own ideas using computational tools, and AI generative tools are some of them. Yeah.
Okay, thank you. So, well, I guess there are a number of questions around AI, and maybe to both of you questions. So, do you know any valuable open source resources to do AI-based drug discovery? And also, what is perspective role of AI in computer-aided drug design?
Actually, in my opinion, I don't find open source AI reliable. There could be many errors in the codes.
So yeah, usually in our case, we develop our AI tools. You may be able to find in GitHub some open access AI, but I think, yeah, there's a lot of interest of these tools, especially Physchem property tools, which are can be based on machine learning approaches. So GitHub, I would say, is the place we may be able to find some open source, but usually we develop our own. or purchase.
Okay, so thank you. And then just one small question to Abdulilah. So during the SCR process, how do you decide and choose which of will be a training or test set compounds?
Actually, there are some rules. You cannot just randomly pick up those training and set compounds. Both sets should cover the range of biological activity. And for common QSAR equations, if you have one parameter in your equation, at least 4 molecules should be involved in your set. And in both training and test set, you should have higher active molecules, intermediate, medium active molecules, and inactive molecules, so that your model should cover all those possible scenarios. Of course, test set compounds are less than training set compounds, but there are no such rules that 80% should be training, 20% should be test. There are no such rules. Of course, training set should be higher than test set of compounds.
That's great. Yes, indeed, there are a number of questions. So, but now we definitely time to conclude. So thank you everyone for attending. Thank you for questions. Thank you our speakers. So we can continue conversation online. Just I guess follow Biochemical Society or Portland Publishing Group on Twitter or X. Yeah. Okay, so we welcome suggestions for future topics and speakers to feature in the biochemistry focused webinar series. So if you have an idea for webinar in 2024, we invite you to submit a proposal for upcoming webinar. You can find more information about the webinars, propose your webinar, and watch previous recordings at the www.biochemistry.org. Okay, so all these webinars are recorded, and this one will be available as well in the next few weeks. And just a little bit about the future. So the next webinar will be on 12th of March at 2 p.m. London time. So, and the next webinar will be Atomic Pathology of Neurodegenerative Diseases. And so we will be joined by Dr. Benjamin Resclidi-Falcon, who will discuss his group's use of cryo-EM to determine the structures of pathological protein assemblies and explore how they guide the development of experimental models. Okay, and finally, I want to highlight that it's important, I guess, than ever to stay connected and engage with your fellow molecular biosciences scientists. And I indeed encourage you to join Biochemical Society community, and this gives you a lot of also benefits, including discount registration fees for society courses and meetings. Also, you could apply for a range of grants and bursaries, and you could have personal access to 2 journals of the Society and Portland Press. So for more information, please visit the website of the Society. And now, I guess, thank you to everyone. Have a great day.
Yeah.